Hello, I'm Professor Heiner Wedemeyer from Hannover Medical School in Germany, and this is Kerstin Stegmann. She's a PhD student at Hannover Medical School, and she's the first author of a paper that we just recently published in Gastroenterology. What is this paper about? Hepatitis C virus infection is a major global health burden, with at least 130 million individuals chronically infected worldwide. Fortunately, we are now able to treat hepatitis C successfully in about half of the patients by a combination of pegylated interferon plus ribavirin. However, we still do not know exactly how interferon works against hepatitis C and what the role is of immune stimulation. In this context, we are interested on the effects of type 1 interferons on NK cells. Thus, in the very first series of experiments, we performed gene expression analysis. We stimulated PBMCs in vitro with interferon-alpha and then looked for genes that were differentially expressed after interferon-alpha stimulation. So Kerstin, what were the results of this first experiment?
We found that TRAIL is among the most significantly upregulated genes, and this finding led us to perform additional experiments. In flow cytometry stainings, TRAIL expression on NK cells increased after interferon-alpha stimulation in a dose-dependent manner. The upregulation on CD56 bright cells is higher than on CD56 dim cells. But how is the TRAIL expression ex vivo? TRAIL expression level as well as the frequency of TRAIL-positive cells is low on both CD56 bright and CD56 dim cells. There are no significant differences between healthy controls, HCV-recovered patients, and patients with chronic hepatitis B or C. This led us to investigate the influence of interferon-alpha on TRAIL expression in vitro. TRAIL expression is increased after interferon-alpha stimulation in the group of HCV-recovered patients compared to chronic hepatitis C patients. Furthermore, we were interested in the TRAIL expression after administration of interferon-alpha in vivo. We investigated TRAIL expression directly ex vivo in patients right before administration of interferon-alpha and on day 2 of therapy. Interestingly, TRAIL expression is only found to be upregulated on CD56 bright cells. Importantly, this upregulation is negatively correlated with HCV-RNA decline. But has interferon-alpha stimulation also effects on the cytotoxic activity of NK cells? Therefore, we incubated isolated NK cells with HCV replicating HLA-7.5 cells. Interferon-alpha had no effect on the viability of the target cells. While resting NK cells only showed weak cytotoxic activity, interferon-alpha stimulated NK cells showed high cytotoxic activity against the target cells. This killing could be blocked in part by adding anti-TRAIL antibody. These pictures are derived from HCV transfected target cells. But what about HCV-infected target cells? Interferon-alpha stimulated NK cells show high cytotoxic activity against HCV-infected and uninfected HLA-7.5 cells compared to resting NK cells. This cytotoxic activity could be blocked by adding anti-TRAIL antibody. We suggest that interferon-alpha-induced TRAIL expression on human NK cells might be associated with control of HCV, thus identifying a novel mode of action of interferon-alpha in the treatment of hepatitis C.
So thank you, Kerstin. So in summary, I think these data show for the very first time a biological explanation for the second phase decline of HCV-RNA during antiviral therapy, which is supposed to be clearance of HCV-infected cells. These data also highlight the importance of NK cell function for the control of hepatitis C virus infection. Thus, it will be important to continue research in this field. We are doing this in our laboratory, and we are also looking very forward to seeing more data from our friends in different labs around the world.